Hello, everyone, and welcome back to the final part of Chapter 13, and where here we will be talking a lot about eukaryotic replication. So, in the last video, we spoke about prokaryotic replication, and now we're going to talk about eukaryotic replication. So, between eukaryotic and prokaryotic replication, the basic mechanisms very similar with each other. It's just there's small little details that differ. And they differ because in eukaryotic DNA, it's a bit more complex than prokaryotic. Because we know in, for example, human cells, there's a larger amount of DNA. There's much more DNA in a human cell than in a bacteria. And we know that they're basically assembled in these linear big structures that we call chromosomes, and they're heavily packed. Because we know if you want to stretch DNA, it can be stretched from here to Cyprus. It's really that big. So we need to really, get these proteins to tightly package DNA in a nice and orderly fashion. So we use, of course, proteins to do so to create these big chromosome structures. And of course, these chromosome structures they're not one circle like this in prokaryotes. In prokaryotes, we know that they're circular, while in eukaryotes they're linear. So these differences is what makes eukaryotic replication different when it comes to prokaryotic DNA replication. So if you look at the enzymology, it's basically very similar. You use very similar proteins or enzymes when it comes to prokaryotic DNA synthesis, or sorry, replication, and the same as basically the ones you use in eukaryotic. Very similar. It's only there's very small differences, and they mostly lie on that you need new enzymatic activity when dealing with the ends. Because in prokaryotes it's circular, while when it comes to humans or basically eukaryotic, it's linear. So you need to deal with it in a different way. So you need new enzymes to deal there. But the rest are basically very similar to each other. They work very similar manner in a similar manner. But one thing about, again, as I was saying, in eukaryotic DNA, it's massive. So one piece of chromosome is really, really, really big. So having one origin of replication might be a huge issue. So DNA can afford that because in bacteria, the DNA is very small. It can afford it. But in humans and human cells, we cannot afford that because it's so big our DNA that if you have one origin of replication, if you want to basically replicate another piece of DNA that's very far down, it might we might not be able to do so because it's so far. It might take ages to get there. So, we need to have multiple origins of replication in each chromosome because the chromosome can be so big, so you need multiple origins. So you have basically, multiple replicons, so multiple pieces of DNA in the chromosome that have a origin, replication origin. And these, regions of or these origins of replications in bacteria, they're very sequence dependent. In eukaryotic cells, they are uh, sequence specific, but they are also structure specific more. So the structure is more important there because if the structure is bigger, sorry, I mean, if the structure is more open, so if this area is a bit more open than the others, because again, remember here DNA is tightly packed. So if you have a part that's a bit less packed and more open, then that can be used as a site of replication. So it's actually the sequence, but also the structure. So bacteria, it's purely mostly the sequence. Humans and eukaryotes, it's actually the structure of the chromosome and also the sequence. And of course, this can actually be adjusted. So during the before the S phase, so before DNA replication starts to happen, you first get helicases. Remember these enzymes? These enzymes were the ones that were opening these double-stranded DNAs, into single strands. So they're basically loaded up, and nothing is happening. They're not activated. They're just there in the replication origins. Then during the S phase, when DNA is signaled to start replicating, you have a subset of these helicases now actually being um, activated, and then you got the rest of the replosome. So the replosome again is this big complex of enzymes all put together to do the replication. So you get all of these different enzymes there to start replication. So for the priming, so remember DNA polymerases cannot just synthesize DNA without a primer. They need a primer. So you have actually DNA polymerase alpha and primase, and they create a primer. So this primer is actually consisting of RNA and DNA. So of course the primase is putting the RNA and the DNA polymerase alpha is putting the DNA. So it's creating this primer that's, a bit DNA and a bit RNA. Remember in bacteria, no, in bacteria it's only RNA. And then you have basically the synthesis. So the synthesis in the leading strand is actually done by the DNA polymerase epsilon, while the one in the lagging strand is done with DNA polymerase delta. So again, same kind of enzyme activity, but different enzymes. So rather than having DNA polymerase 1 and 2 and 3, here you have DNA polymerase, for example, epsilon or delta. And of course, as we see, the replication of DNA is actually very similar among the three main types of domains of life: bacteria, eukaryotes, and archaea. But if you look actually really properly at the enzymes, if you look at basically their structure and their homologous domains, surprisingly, archaea and eukaryotic replication proteins are evolutionarily very related. They look so similar, specifically DNA polymerase, replicative helicase, and primases. They look so similar to each other, and they're a bit different than the ones in bacteria. So most probably what happened through evolution is that eukaryotic and archaea cells actually, kind of together went through this evolutionary kind of process of developing very similar proteins for DNA replication, while bacteria went in a different path. It got split somewhere else, and they have different enzymes that look different. Of course, they work in a very similar manner, but their structure and their domains are different from each other. So again, eukaryotic and archaea, these enzymes, DNA polymerase, replicate helicases, and primases, are very similar, and they're different to the ones in bacteria. So one thing that I also said at the beginning is these um, ends. So the ends of a eukaryotic DNA, it's linear. It's not circular like a bacteria. So we need these special structures called telomeres to help us actually with these, ends, these kind of linear ends. And these actually are just structures that are composed of a specific sequence. And we'll talk about them much more. So imagine this is basically your DNA. So basically you have here in green, this is your leading strand, and this is basically your lagging strand. So remember the leading strand is the one that's facing 3' to 5', because when you add DNA, it's going from 5' to the 3' end. So this is perfect. There's no issue with it because it's going continuous in one fashion. But when it comes to the lagging strand, as we said, you create these multiple Okazaki fragments. So we're putting a primer here and a primer there, and then basically doing the synthesis there and there. So creating these Okazaki fragments actually. So in this case here, when basically this DNA polymerase delta comes in, it's going to remove these primers and it's going to add technically now these new formed nucleotides and everything. So you're going to remove them and then it's going to be able to assemble new DNA nucleotides. Here, it's able to do so because it uses this piece, this Okazaki piece, as your primer. But here, when you remove it, the polymerase is not going to be able to do anything because, again, it doesn't have any primer here. It cannot use this primer. It needs a primer basically, up behind it, but it doesn't have it. So basically, what happens? You remove the primer. This thing now cannot be done, and in generally, this actually is removed. And what happens is that this now chromosome basically is shortened. This piece of DNA is shortened because this is gone. This primer that has the sequence has been removed. However, in eukaryotes, this doesn't happen because we have certain enzymes called telomerases, and telomerases take care of these ends. So what these guys do is they're these big, massive, complexes of enzymes, and they actually have, so this is a telomerase. It actually has this internal RNA template. So this is the internal RNA template that's able to actually bind to the ends here in the lagging strand. So it binds to it, and what's going to do is going to add more actually nucleotides, and then it's gonna go back and it's gonna again add nucleotides. And as you can see, it adds a specific sequence, this repeating sequences. So you see here T T G G G G T T, and then basically, so you have this repeating of T T G G G G, and then again you repeat it again T T G G G, and it does it into multiple times until basically the ends are then corrected. So this is what's preventing the ends from basically not being cut off. because this part you have this telomerase that has its own internal RNA primer that's able to actually, bind to this part here, and it's able to extend it. So you create these telomers. So the ends of these chromosomes are actually have this specific sequence, these telomere sequences that are literally repeating on and on and on because of the activity of this enzyme, this telomerase. So this is what actually protects it. So telomerase activity in development is regulated. So during, of course, development in childhood, where our cells are multiplying quite constantly, and of course we need to replicate DNA, the telomerase activity is required because imagine if it's not there, we would have basically these ends being cut short and short every time DNA was replicated. So sometimes these chromosomes could disappear after multiple replications. So we need this to be quite high. Then later on, during adulthood, actually it starts to decrease. You start having low amount of telomerase activity, and specifically in somatic adult cells, in cells that do not actually replicate. In other cells that keep dividing a lot, for example, like your immune cells that replicate quite constantly, no, they're actually there. So you have some exceptions, but most of the somatic adult cells have a very low telomerase activity. But what they did is they actually saw a very cool experiment and where they actually got mice and they removed telomerase. They removed this enzyme, and then what they saw is that after 6 generations of breeding, they couldn't create any more actually viable offspring. They were not able to do so because their chromosomes were almost kind of cut together. They were being shortened so much that certain DNA that really was needed for life was not there anymore. So you start to see something that senescence, which is also aging of the cells, is actually correlated with telomers. So a cell that is actually, getting older actually has smaller telomers. So these small, basically, structures at the end. So the chromosomes start to get smaller and smaller because these telomers are basically going away. And then one thing they start to see is that in cancer, these somatic cells that are, constantly actually, replicating, they sounds like, but why are they replicating? Aren't they supposed to have their telomeres actually go smaller and the chromosomes start to actually go down? No, they saw that cancers generally have a huge activation of telomerase, and that's what's keeping the telomere length quite still and they're intact, and it actually helps DNA replication to keep going without the chromosomes getting smaller and smaller. So you can see that telomerase plays a big role when it comes to aging and in cancer. And of course, these are the ones that discovered actually more the telomeres and telomeres activity. Elizabeth Blackburn, Carol Reeder, and Jack Szostak, and they actually were their discovery was so important that they actually were given the Nobel Prize in medicine, which is a huge prize. It's the most prestigious award you can ever get as a scientist, and they of course were really well deserved for their contributions here. So one thing now, my final thing that I want to talk about is more about DNA damage. So DNA is constantly, constantly actually, replicated. And sometimes what happens is that there are errors that are happening. The DNA polymerase is sometimes, adds certain, errors there. But also our DNA is constantly actually also exposed to many different mutagens. These are agents that increase the number of mutations. So mutagens can, for example, be UV light. They could be smoke, be certain food additives that actually cause DNA damage, and they can basically damage the nucleotides in your DNA. So these are mutagens. So DNA has to have some sort of way to repair this, to repair the errors and repair these mutagens, causing damage. It needs to, and there's multiple types of repair systems. And here today we're going to go through a small amount of them. So there's two categories of DNA repair: the specific repair and non-specific. So specific repair means that you are actually fixing certain lesions of DNA and only repairs a specific type of damage that we'll talk about. Non-specific, it doesn't care. It will actually, repair multiple kinds of DNA damage. I will go more into specifics about this. But I want to go just before I go into these different types, I want to talk about this mismatch repair. As I said, there's a lot of errors during replication, so we need to make sure that we correct this. So basically, the DNA polymerases have their specific machinery. So this mismatch machinery that's able to actually recognize that okay, DNA polymerase added some sort of mistake. We need to correct it. So as you remember, these mistakes are added to the newly synthesized strand. The template strand doesn't have anything, so we need to make sure that this mismatch repair mechanisms are able to distinguish between the template strand and the newly synthesized strand, where actually the mutation or the incorrect nucleotide added is in the newly synthesized strand. It's able then to basically, recognize it and repair it. So in E. coli, this actually involves the methylation of A in the sequence GATC3. How is it able to actually distinguish it is that the template strand in E. coli is methylated. The A is methylated in this sequence, this GATC. In the newly synthesized strand, it's not methylated for a short time. For a short time, it's not, and this mismatch repair system has that small window to go and actually distinguish. It's like, okay, this is actually the newly synthesized strand. This is the one that I need to actually repair because the newly synthesized strand will not have this unmethylation for this brief window of time. So this is how you are able to actually repair errors due to replication. When it comes to, for example, specific repair mechanisms, you have one that's called photo repair. And what happens is that UV light loves to basically, attack thymine. So thymine, what happens is when it's attacked by UV light, it starts to create these thymine dimers in a way that, it will bind to an adjacent thymine rather than it's basically adenine. So you create these dimers, this together, and this is a huge problem because it can create a massive problem when it comes to DNA. If this DNA is actually then expressed into a protein, the protein can be very incorrect. So we have these big proteins, basically these photolyase proteins that bind to the damaged DNA, and it absorbs light. This photolyase absorbs light, and it gives it energy to repair this, so to break this bond between these thymine dimers and correct it. So this is how this is specific, that this photolyase can only actually correct damages of thymine dimers, which is caused by UV light. So it's very specific to that. You have something else that's called excision repair, and this is non-specific. So non-specific repair here, it doesn't care what type of damage it is, it's going to do so. And it does it through three very simple stages. First is recognizing the damage. What is damaged? Then it removes the damaged region, and then it basically signals for resynthesis of this undamaged strand, of this damaged strand. So, for example, here you have some sort of happening, so either damage or an incorrect base. You have this big complex called this UVR ABC complex that binds to damaged DNA. So this is the first step. Then it removes it, it excises it, it removes it, and it calls this DNA polymerase to come and repair it, basically to repair the damaged strand using actually the undamaged strand as your template, and it's able to do so. This in this case is DNA polymerase, and that's how you use excision repair. So this is the end of this basically chapter. Good luck with it, and thank you.